recording because we have several students that are out with the flu, so it's going around. So be real careful, wash your hands a lot, kind of limit contact, things like that. I knew it was going to come. I knew it was going to hit pretty hard. Okay, so let me get all this out of the way. I'm going to share on the screen. Yes. I'm just checking something here. There we go. Okay, we're definitely sharing. Okay, so Tuesday lab, I'm so sorry. I forgot to hand back your previous lab quizzes. So they're in here. I just was so, focused on getting you going and everything like that and review. So that's in here, including anything else you haven't picked up. And then of course the sun. So yeah, regarding this flu, it's definitely going around. The timing is terrible because we're coming up on Thanksgiving break and you don't want to be sick on break. Right? That's and get your flu shot. So, so I've got this up here. I've got a couple of announcements other than the flu, which has definitely been a concern. So we're on track to finish this chapter by Friday, definitely. Okay. And then of course on Monday is our exam and no labs next week. And then we come back from that break and we will have our final lab of the semester, that case study, which is voluntary. I mean, you don't have to come if you feel like your 6 lab quiz grades are fine. You don't have to come to that lab. All right. That is just for people who want to get a shot at doing better on these 15 questions than one of their lowest lab quiz scores. And then we can drop that. All right. And then the week we get back until the, I guess, last day of class is the reproductive physiology. And I got that PowerPoint up. So trying to get our ducks in a row, but the end is near. The end is near. Yeah. I just feel like you guys have kind of lost a lot of steam. You don't want to let up the momentum now because you still have this exam coming up. You have the final exam. You have a final lecture quiz, all right, which is going to be respiratory, I believe. So, all right, I've actually got the final exam already written. I've got an exam breakdown. So basically, this day, I'll probably email that to you. And our exam, our final exam will be Friday of that week, but at 8:30. But anyway, I want to encourage you. You've made it this far. Don't let up the steam and momentum now. Now is where you just got to push yourself. If you got to keep chugging, power drinks and coffee and espressos and all that stuff, do it. But you're almost there. So I want to encourage you because I noticed I felt like a drop in energy. All right. I think you all feel it too. And I feel it too. I'm just trying to keep myself up to keep you guys motivated. All right. Remember the end goal you have. Get into the nursing program. All right. So you want to make sure the best thing you can do is get highest scores you can get to make sure that happens for you. All right. Okay. Any questions about any of that? Yes. Well, I have to keep it a surprise. Because and for the case study, if you decide to come to that lab, okay, which is the lab after Thanksgiving, bring your smartphone, your iPad, your computer, because you're going to be doing web searches. That's what the lab is designed to do, and you work together as a table for the mystery question. Right? You're going to basically diagnose a disorder based on a patient's presentation, and then you're going to look at treatments for that disorder and try and explain why those treatments are prescribed for that disorder, and then talk about side effects and very clinical, very relevant. The nursing program, the farm program, they have these kinds of case studies. In fact, I showed it to Dory Weaver. It's just, that's a pretty good case study. All right. So I want to kind of give you a feel for that as our last lab. All right. So bring your computers, use them. Last thing before I get started is I've been going back and forth with Corporal Carmichael about these CPR cards. All right. And so I sent him, you replied to my email saying, if you haven't received your card, email me saying, haven't received my card. And so I made a list. And then I copied and pasted your email address and I sent it to him and had him double check his original email list that he sent to PD Regional. They're the ones that are issuing the cards, sending out the email to you. And he found 3 discrepancies where the emails weren't a match. And I just emailed those 3 people out of 12 people. He said they didn't receive their card, or he's notified through colleague I think, whatever Patriot portal, when students haven't received their card or when they've received it. He said there's 12 people that he saw didn't claim their card. And of those 3, there was an email discrepancy. And so he fixed it. And then you should be receiving that email from PD Regional with the link to claim your card. And those 3 people that had the discrepancy in the email, I emailed you just before lecture today. The remaining people I email on my list, because you contacted me, I said, okay, he's checked your emails and they're correct. So check your emails again, check your spam folders, wherever else something could have gone. And then at the end of that email to Corporal Carmichael, I said, is it possible that PD Regional could send me the email with the link for the students who haven't received their card or haven't received their email? Because I had no problem receiving it. It went to my spam folder, but I looked in my spam folder, it was right there. Okay. And I received my card like a couple days after the lab. All right. So I've tried all these options to try and solve it for you guys. And so I'm kind of waiting to hear. Check your spam folders or wherever else things go. Yeah, I mean, I think that's all I can do. All right. So I'm working on it. I'm not letting it drop. So I'm not letting the ball drop. Don't you guys let the ball drop. Okay. All right. Now, are there any questions? Okay. If you come up with one, just email me. Okay. Okay. So I think we left off with this. Okay. So we are still at there. We are still at when you're at innate immunity, which is what you're born with. It doesn't involve antibodies. And this is once the pathogen enters your body. So this is called internal innate defenses, whereas last time I covered your barriers, your skin, alveolar macrophages, acidity of the stomach, acidity of the urethra, vaginal, all that stuff. That was barriers. Now something got in. And so we talked about what white blood cells do, and then we talked about what natural killer cells do. And now we're going to move on to your inflammatory response inside, once something has gotten inside. So there might be an exam question about this, because white blood cells come under blood physiology, which is chapter 7. But mast cells are basophils. Oops, changed my pen color. They are basophils. Okay, so we talked about neutrophils, lymphocytes, monocytes. I'm going through never let monkeys eat bananas, eosinophils, and then the bananas, the most infrequent cell, the lowest frequency, is basophils. All right. They secrete histamine. And I don't have room, so I say, which causes inflammation in allergic reactions and immune response. I mean, really, an allergic reaction is an immune response. You come in contact with something, all right? You get stung by an insect, all right? And when it swells up and gets red and feels hot, that's a response of histamine, all right? That's the inflammation. When your nose runs and your eyes run, that's a histamine reaction. And it is inflammation, your eyes get all red. What your body's trying to do is flush with tears and everything like that. Flush whatever antigen, pathogen was exposed to your eyes and get it out of there. Okay. So we all know a histamine reaction, and then taking antihistamines is supposed to bring that inflammation down. Okay. So that's a common response of your body as an immune response to protect you. Okay. And also the heat that can result can also be a high enough heat to kill bacteria. Sometimes it works, sometimes it doesn't. Complement proteins, I'm not going to talk much about those, but their job is to destroy bacteria. By, let's see if I can get away with space here, poking holes in them. All right. So there's like 40 complementary proteins, and I'm not going to go into all that. Very complicated, but really the job is to destroy bacteria. They poke holes in them, and then water rushes in and they lyse, all right? They swell up and they burst. And so that's what happens with complement proteins. So again, this is all, they also cause inflammation. Both of these cause inflammation, right? But I'm trying to distinguish between, yeah, these both cause inflammation, but this one is different in that it secretes histamine. This one is different in that it destroys bacteria by poking holes in them. Okay. So this is a mast cell or a basophil that is actually secreting the histamine that causes the inflammation reaction. And since I see that I wrote over the image, and if I post this later on in the week, you're not going to be able to see it very well. So I'll try and move this image up so you can see what I wrote. All right, I'm trying to do that. Okay, so we're still on phagocytic white blood cell response. So what I'm going to do is kind of like a stepwise thing. Remember how in the review session in lab this week, I talked about for chapter 6, muscle physiology, I did the sequential question that is on the exam. There's no doubt about it. I wrote it, of what happens from when a neuron releases acetylcholine to when a muscle contracts. For this chapter, you're going to want to know the sequence as well. All right. But for the final exam. Okay. So the first step, once something gets into your body, okay, the first responders are going to be neutrophils, and then monocytes come in pretty soon too, because never let monkeys, they're pretty common. They're phagocytic cells, and I'll move this later on so you can see what it says, that destroy pathogens. Phagocytic cells, did I write it down before? They eat pathogens. I think I wrote it in an earlier slide. Phagocytic cells, like your white blood cells, especially these two, consume pathogens, endocytosis, and then they destroy them with digestive enzymes. They also secrete endogenous pyrogens to cause the fever. I wrote that down in an earlier slide. That's what a neutrophil looks like. That's what a monocyte looks like. Okay. Now, histamine secreted by mast cells, basophil cells. What histamine does, and remember back when I was talking in the cardiovascular chapter, I was talking about shock. And I was talking about anaphylactic shock. And I said, for all forms of shock, what you have is a massive systemic drop in blood pressure. And I think I mentioned, probably didn't write it down, that what happens in an anaphylactic reaction, it's an over exuberant immune reaction. And what will happen is, normally when you have a mild allergic reaction, you have some histamine release. In an anaphylactic reaction, you have a huge histamine release. And what that does is it makes your blood vessels leaky. And I have a little GIF that shows that, so that monocytes in particular can escape your bloodstream and migrate out of that blood vessel into your tissues. Because if the pathogen's not in your bloodstream, what happens if it's in your tissue? So it allows them to escape your bloodstream and get into tissues. Now, that's great, because then that monocyte can wage war on a pathogen in your tissues. The downside to your blood vessels being leaky is they'll actually vasodilate as a part of this histamine reaction. And that's why in an anaphylactic reaction, which is a lot of histamine, you have a systemic drop in blood pressure. All right. It's too much histamine. It makes your blood vessels dilate. So I'll put blood vessels leaky. Oop. Let me go back. Makes your blood vessels leaky and vasodilate. By vasodilating, that's what helps them become leaky. Now, the white blood cells, specifically monocytes, can escape your circulatory system and get into your tissues. So the whole thing is to get that monocyte out of the bloodstream into your tissues. So that's the first response, is they're in the bloodstream. The next response is monocytes escape. It's called extravasation, out of the bloodstream into the tissues. And now we're going to talk about what that monocyte does from that point on. It's going to lead you down the path of pathogen got into your body to antibodies being made. That's the sequence. All right. And this process takes 2 or 3 weeks of time to happen. All right. This is why when you get a vaccination and it's just something new, like when you got your first COVID vaccination, they said in 3 weeks, come back for a booster, because they're expecting you won't get antibody production at all until probably good antibody production until around 3 weeks. this process I'm explaining to you is that 3 weeks. All right. So is it relevant to nursing? Yeah. Okay. So this is extravasation. All right. This is the endothelium of the blood vessel. And this is the monocyte that just squoze out of that blood vessel into the tissue. Oh, and influence of histamine. I'm not going to get into the influence of serotonin. It's simply too much. So histamine allows this to happen. Now, from there, all right, the monocytes leave the bloodstream. They extravasate to leave out, and they become this thing. They become a macrophage. Okay, and I said macro is a big eater, cell. I know I wrote it down someplace else that the macro means big and phage means to eat. They're big eater cells, like Pac-Man. Now they're migrating through your tissue, and the little dots are pathogens. It could be bacteria, it could be something else. All right, it's going to do something else very important. When it comes in contact with that pathogen, it's going to become something else entirely. And it's going to start the communication between innate immunity and adaptive immunity. Innate protection without antibodies, adaptive immunity, protection with antibodies. So this interaction right here is going to be vital. So the macrophage, it will destroy the pathogen, okay? It consumes it, breaks it all apart. But on the exterior of every pathogen, let's say it's a little bacterial cell, let's say it's a little rod-shaped bacteria. It has little surface antigens on it, all over it, that is its identification, okay? So when the macrophage destroys this bacteria, let's say, it keeps these antigens and it presents it on its surface. So the macrophage takes the pathogen's antigen, and now the macrophage has become something new. It's called an antigen presenting cell. And I just abbreviate that to an APC because it's easier. It's like a waiter that's serving you food, all right? Comes out with the dishes on their arm. The macrophage is now an APC. It is carrying on its surface an antigen. And it's looking for a cell of adaptive immunity to give it that antigen and say, this is the bad guy. I want you to make antibodies to this. So this is the link between, right here. An APC is an agent of innate immunity. And it's going to activate an agent of adaptive immunity called a helper T cell. All right. This is part of adaptive immunity. Okay, that's the link. And then this is gonna, the helper T cell is the most important T cell in your immune system. And it's activated by an antigen presenting cell. So the antigen presenting cell is like this. Hey, we're a helper T cell. I need you. I got an antigen for you. Helper T cell comes along, is exposed to that antigen, and then it's going to take it to your lymphocyte cells, which make antibodies, and say, here's the antigen, make an antibody to it. So when I talked about before, and I have before, T cells being the most important part of your immune system, specifically talking about helper T cells. When a patient who's been exposed to HIV has human immunodeficiency virus, okay? And that virus has antigens on its surface too, but that virus is really good at hiding out in your immune system and basically pecking away at your immune cells. Basically, it's destroying helper T cells. And when T cells fall below a certain threshold, they will diagnose you as now have gone from having HIV to having AIDS, acquired immunodeficiency syndrome. Why is your immune system deficient? Because these helper T cells have been destroyed to the point where your immune system can no longer function. This thing right here runs the whole show. Once it's exposed to that antigen, it is a bossy T cell. It says, okay, I want you other immune cells to rush to the site and mount a defense. I'm going to send out chemical cries for help. Hey, B cells, I want you to make antibodies to this. Now, get on. And it's directing everything. And if you lose those T cells, your immune system's done. All right. So that's the importance. This is communication between innate immunity and adaptive immunity, and this sucker runs the entire show. All right, so that's his importance. Okay, now the phagocytic cells, the macrophage, which has become an APC, and any other cells also secrete chemical cries for help. In fact, all white blood cells can do them, mast cells can do them, macrophages can do them, they all can do this. So they're sending out the alarm, woo, woo, that is bring all the immune cells to this site. This is where the war is being waged. Okay, so this is a macrophage that is consuming a pathogen, destroys it, teases apart its antigens, and now wears it on its surface, and is now going to be waiting for a helper T cell to come along and take that antigen and then just take over the show from there. Okay, so that's kind of what it looks like. Okay, so here is a helper T cell that is getting the antigen from an antigen presenting cell. And now that the helper T cells become activated, it takes over. It does everything from there. All right. And so I have a slide much further down showing all the things that this helper T cell does. All right. So that's the impact. That's what I wanted to go over today, is that helper T cell. Okay. So we are still in innate immunity. All this was showing you macrophage leaves the bloodstream, monocyte leaves the bloodstream, becomes a macrophage, then it encounters the pathogen, wears it on its surface. It's now an antigen presenting cell. That is still innate immunity, but that is where we have an interaction with adaptive immunity. But I'm not done with innate immunity, so let's continue on. Okay, we'll come back to T cells in a bit. Natural killer cells, I had this on another slide. I have it cartoonish because this is how I think about it in my brain, but natural killer cells of innate immunity secrete interferon to destroy viruses, all right, and toxic granules, blam, all right, to kill tumor cells, which aren't cancerous yet, but could become cancerous. All right. So we have all these checks and balances in our body to protect us. They are activated by the cries for help. So they come to the site, and if it's a virus, release interferon. If it's a tumor cell, secrete the toxic granules. So I've got, here's a tumor cell, and it's going to encounter a natural, here's a virus. See, it's got the spikes on its surface. Here's a virus. And then the interferon secreted by the natural killer cell made it get destroyed. All right, so let it run. Virus, all right, does something with the interferon, virus is dead. All right. I tried to find videos of the toxic granules in tumor cells, but it was very difficult. Okay. Repeat, this is still repeat. Inflammatory response, I already covered this on another slide. I don't know why I have this here. Oh, I know why. It shows more of histamine released by the mast cells, but here it shows basically a rod-shaped bacteria with holes being poked in it by the complement proteins, so that water rushes in and the bacterial cell ruptures. That's the only reason I have this slide here. All right, so it's the same information. Mast cells secrete histamine, all that inflammation, your blood vessels dilate, your blood pressure will drop a little bit, but that allows the monocyte to leave your bloodstream to become a macrophage, to become an APC. Okay. So this is a picture that kind of has all the events and happening, starting with here's your bloodstream, and this is in the tissue, and here's your skin. So let's say you get a cut in your skin. Bacteria, again, rod-shaped bacteria are entering. So we have white blood cells in your bloodstream, neutrophils, monocytes. If it's in your blood, the bacteria, they can do their job. But if the bacteria is in your tissue, you need those white blood cells there. So what happens is they attack, they secrete endogenous pyrogen, they start a fever response, they send out chemical cries for help, okay? And the next step is monocytes leave the bloodstream, okay? Because of a histamine reaction, blood cells, bloodstream, the vessels become leaky, and then the monocyte can leave through extravasation, and then it becomes a macrophage when it's in the tissue, all right? And the next step is the macrophage eats a pathogen. So here it's attacking the rod-shaped bacteria, taking it in. And what they're not showing here is the antigens end up being on the surface of that APC, antigen presenting cell. All right. And what's it waiting for now? Okay, the APC activates T cells, helper T cells, the most important part of your immune response. Okay. The T cells, I'll get to it soon, takes over everything. But the APC also secretes chemical cries for help. They all can do it. So more cells come in. So in this particular picture, we don't happen to see the helper T cell anywhere around, but imagine it's on its way. Here we see a mast cell secreting the histamine, which makes the blood vessels leaky and causes swelling and redness at the tissue where the break was. In this scenario, we already have a B cell, a B lymphocyte that has antigens to this bacteria. In this scenario, this person has already been exposed to that particular bacteria and has antibodies ready. And so with the chemical cries for help, these B cells come to that site and start chucking their antibodies at that bacteria and binding it up. So do you know how antibodies work? You kind of do now because you have the blood typing lab. An antibody is this Y-shaped thing. So the B cell is not itself attacking the pathogen. It's chucking its antibodies at it. Look what's happening here. The antibodies are binding to the pathogen or the foreign antigen, and it's sticking to it. And then other bacterial cells that have the antibodies stuck to them, those bacterial cells will get stuck together. When cells have been bonded to an antibody and they get clumped together, what was the term I talked about in lab this week? It begins with an A for that clumping process. The little flecks you saw in your wells where you had a positive reaction, you added antibodies to your blood. And if your blood had the antigen to which that antibody was prepared for or specific to, the antibodies clung to those antigens, those cells, and they stuck together. What's the term for the sticking together? Agglutination. That's what happens with the bacteria. The antibodies stick to them, and then all the other bacterial cells, and then they start to clump together. It's an agglutination reaction, just like you saw in lab with the antibody you added in your blood. Your blood had an antigen, and that antibody bound to it. This is actually how your immune system works. This is just chucking the antibodies. Once the bacteria are all stuck together in an agglutination reaction, they can't migrate, they can't move around, they're stuck, they're all stuck together, right? And then they're at the mercy of all the other immune cells of the body. They come migrating to the site because of cytokines, chemokines. More macrophages come in, more mast cells, all this, and they start to consume all that bacteria and destroy it. Once they're stuck together, they're at the mercy of your immune system, and then your immune system wipes them out. So when they're agglutinated, they can't escape. That's how your immune system protects you. Anyone actually know that? Ever hear that before? Okay, I figured it was going to be new to you, but that's what happens. Okay, the mast cells are releasing the histamine, redness, swelling, making the blood vessels leaky. Okay, cytokines, chemokines call all these cells, especially B cells, B lymphocytes that have antibodies to that site. Now, if you don't have antibodies yet, then the macrophages and other cells will come in and just try and destroy the pathogen as best they can. All right, in this case, we already have antibodies to that. Okay, so we've covered a lot. We have previously covered this, and today we get to this, and this. And we also started the sequence. Pathogen enters your body. White blood cells in your bloodstream mount war. Histamine released by mast cells makes your blood vessels leaky. A monocyte can escape into your tissue. It now becomes a macrophage, big eater cell. It eats the pathogen, puts its antigen on its surface as an antigen presenting cell. And then there's all these other cells of innate immunity that I talked about. All right, so from here, we're going to be moving on to what happens now that that T cell is activated. Okay, adaptive immunity. Yes.
If we take antibiotics every time that we get sick, do we reduce our body's chances of producing their own antibodies?
Repeat it again.
If we use medication to treat the bacterial infection, do we reduce our body's chances?
Are you talking about antibiotics?
Yeah.
Okay, so if you use antibiotics, do you reduce the chance of your body's ability for forming its own antibodies? No, but you do something else that's very dangerous. Okay, everyone get that? She asked if you take antibiotics for a bacterial infection. I mean, if you've got a bacterial infection and they measure your blood and your white blood cells are sky high, leukocytosis, all right? yes, you have to take antibiotics because it's gotten to the point where it's kind of escaped your immune system and now it's causing a problem, especially if you've got a fever. If it's just a mild infection, remember I said mild increased fever is okay. Should be able to kill that bacteria, but it's when it gets bad, absolutely you must take antibiotics. Yes. Okay. Now, if it's a viral infection, are antibiotics going to do anything to that? No, antibiotics are against bacteria, not viruses. You have to hope that your own body can protect itself, or if there's a vaccine available, you take that and you boost your immune system's ability to produce antibodies specific to that virus. Antibiotics don't do crap to a virus. Okay. The problem we have, and it is a big one. Does anyone know what the problem with this when people get prescribed antibiotics for a virus? Please tell us, Zachary.
Virus. Antibiotics, so if they start getting less effective, it's bacteria.
Bacteria can become resistant, but that's not the big problem we're facing. Well, overuse of antibiotics, especially when it's prescribed to treat a viral infection, which happens a lot. People take their kids and say they've got a sniffle and they want them immediately on antibiotics. What if it's a flu? What if it's a cold? The antibiotics aren't going to treat that, but now that person has been exposed to this antibiotic for no reason, possibly. Okay. And when that happens a lot, bacteria, which reproduce every 20 minutes through mitosis, they contain genes for resistance to that antibiotic. And when the bacteria are exposed to that antibiotic a lot, you start to breed forms of bacteria that are resistant to the antibiotics that you have to treat it. Antibiotic resistance is a real problem. Okay. There's antibiotic resistant E. coli. There's antibiotic resistant gonorrhea, which is bacteria. There's antibiotic resistant all kinds of organisms. All right. There might be some forms of antibiotic resistance to flesh-eating bacteria. When you overuse antibiotics, especially if you treat it for a viral problem, you're just exposing someone's immune system to the antibiotic, and any bacteria that are in their body can become resistant to it. So does that help explain it? Antibiotic resistance is a big problem. A big problem is prescribing antibiotics for viral problems. You're not doing anything. Now you're exposing someone's body to an antibiotic and you give bacteria a chance to become resistant to it. Google it, WebMD. It's a real problem in the medical community. All right. So we're trying to devise new antibiotics. Because it didn't even occur to me. Let me get my connection back. It just went to sleep on me is all. Let's get back in here. Hopefully I'll be able to get back in. And it looks like it's going to let me back in. Let's give it a sec. There we go. Okay. So here we are. Adaptive immunity. Sorry for the technical glitch, but it was a question so worth asking, so worth answering and explaining that. Okay. Adaptive immunity. It's going to involve antibodies, but not all the cells of adaptive immunity produce antibodies. T cells don't produce antibodies, but yet they're a very important part of your adaptive immunity. So your lymphocytes, okay, are part of adaptive immunity. They're produced in the bone marrow. T cells are T lymphocytes. B cells are B lymphocytes, okay? T cells mature in the thymus. B cells mature in your lymph node spleen and other places. Okay, so T cells. Helper T cells, cytotoxic T cells, regulatory T cells. There's 4 types of T cells that I know of. And there's 2 types of B cells. All right. This is actually going to be much less complicated than innate immunity. It's actually pretty straightforward here. Once you activate helper T cells, it's actually pretty straightforward. All right. Okay. So T cells are part of cell-mediated immunity. So when it says cell-mediated immunity, it's T cells. If it says antibody-mediated immunity, it's B cells, because T cells cannot produce antibodies. So I'm afraid if I turn this around, I might lose my connection. Let's do it anyway. So T cells do not produce antibodies, do not themselves make antibodies. The only ones that can do that are your B cells. All right, so there's a difference between them. But next I'm going to go over, while I have some time, the four different T cells, I believe, in what they do. Okay, so this is again, I've already gone over this. An antigen presenting cell, it's becoming one right here. All right, so it's got the antigen. It's going to be looking for a helper T cell. And when it encounters it, here's what happens. Again, like a comic, it's what I think about. So here we have a macrophage, okay? Oh, it's not doing it for me. A macrophage consumes a bacterial cell. It then puts part of that antigen on its surface. It's now an APC. All right, so here we have an APC. Hey, now I'm an APC. Here you go, helper T. The helper T gets that antigen. Okay, says, oh, thank you, APC. Now I'm activated. And it's going to take over the show from here. All right, so that's innate immunity. This is adaptive immunity. All right, so that's the important part. That's why in AIDS patients, they've lost their helper T cells to a level that they can't protect you anymore. Okay, so here we have the categories. Cell-mediated immunity is T cells. Antibody-mediated is B cells. So helper T cells get their antigen, get activated by an APC. It becomes activated, and then it activates all the other T cells, and it activates the B cells to make the antibodies. So once it's activated, it takes over. It will now activate other T cells, cytotoxic T cells. They're also called killer cells, but I'm not going to call it that because then you'll get confused from natural killer cells of innate immunity and then the cytotoxic T cells of adaptive immunity, and I'm not going to do that. So, and I abbreviate it with the T subscript C. They kill pathogens on contact. Okay. The helper T cell has the antigen, right? Helper T cells activated by APCs. Okay, helper T cells have the antigen. They have the identification of the pathogen. They activate cytotoxic T cells and say, hey, here's this antigen. You find it, kill it on site. And that's what they do. Okay, the next ones are memory T cells. They retain, it kind of redundant, retain a memory of the antigen. Okay, that's their role. And then regulatory T cells are something a little different. They actually, for every action, there's usually an equal and opposite reaction. Regulatory T cells inhibit cytotoxic T cells and B cells, which make antibodies. Why would you have a cell that does that? Okay, and this is relatively new information. I mean, they're discovering this stuff all the time. They think the role of these cells is to guard against anaphylactic reactions or severe allergies, but the big thing is autoimmune disease. What's going on with autoimmune disease? Your own immune cells are attacking your own tissue. And so these cells are thought to prevent this. And so a lot of researchers now, and it'll come out maybe in the next 10 years, are looking into, are the regulatory T cells not doing their job? Are they just not doing their job in people who have autoimmune disorders? Can you imagine if they figure this out and they know how to reactivate these regulatory T cells that are having problems, that people who have autoimmune disorders, they won't have to take immunosuppressants anymore, and they won't have to take these Humira. It's one of the most commonly prescribed, basically immunosuppressants out there for people who have Crohn's disease, who have inflammatory bowel disease, who have whatever autoimmune disorder you can think of. And Humira is hard on the body. Do you know anyone who's on it? I know one person, and they say when they get their Humira shot, they're done for the day. She's like, that's it, I'm out, I'm done. Don't email me, don't contact me, I'm done for the day. It's hard on the body. It makes you prone to getting flus and colds because your immune system is suppressed to keep the autoimmune disorder in check, but it has all these side effects. If they can figure this out, you don't have to worry about that anymore, right? But that is kind of new on the horizon. Okay. Helper T cells also activate your B cells, okay? These are the ones that make antibodies. Antibodies are known as immunoglobulins. In nursing, you might learn all the different immunoglobulins. There's like, when I took biology, I learned of like 4. with Greek letters and all that stuff. But anyway, these are the Y-shaped antibodies that go after the antigen. You saw in lab, when you add antibodies to a Petri plate with your blood that has an antigen for that antibody, clumps it. All right. We have two types of B cells. Let me move this, it's in the way. Put it up here. You have, come on, pen, plasma B cells and memory B cells. Okay, I'm going to put here plasma B cells. You're not going to know. Put up your dukes. Such an old reference, but I always remember it. This means do this. Get ready to fight. Get ready to fight now. All right. Put up your mitts. Okay. Ready to fight now. So as soon as they're made, as soon as these plasma B cells are made to the antigen that the helper T cell gave them, they're being pumped out by your lymph nodes and your bone marrow, and they're ready to fight the pathogen right away. All right. Now, the body also makes memory B cells that wait in immune system, in your lymph nodes and all that, for later, to fight later. Oops, I'm running out of space. To fight later. Okay. Now I think I have this, I have these little GIFs here that show it. In talking about this, you have B cells that can fight now. When you are first exposed to an antigen your body's never seen, like a COVID vaccination, you're exposed to this something that's supposed to stimulate you. They give you a part of that virus. And everything happens in innate immunity. Helper T cells they are exposed to this, and they take it to the B cells, and you make plasma B cells that are ready to go now. That first vaccination and that 3 weeks it took to make these and these, they're ready to fight now. But your protection is not very, it's okay with one shot. Okay. You have some plasma B cells, you have memory B cells being made, and they're waiting. When you get that booster, you have now so many memory B cells that were produced from the first shot and that get activated from the second shot, because the second shot is a second exposure. All these memory B cells that were waiting, when you get that second exposure, that booster shot, holy crap, there's a huge difference in your protective immunity. First shot, when you make antibodies, okay, you got some plasma B cells, they're ready to go. All right, second shot, all those memory B cells that were formed in the first shot, they descend, and you've got huge protection. Does that explain it to you a little bit? I have something here that, okay, here we have the helper T cell being activated, all right, by your antigen presenting cell. It becomes activated, okay? This is basically showing you what happened in lab, but this is bacteria. Your B cell, and I'll just say plasma B cell, chucks its antibody at the bacteria. It gets stuck to it and all the other bacteria, and they clump together. And we have, let's say, a cocci-shaped bacteria. They clump together in agglutination. And you saw that yesterday, which is really nice because you can actually see it, how fast it happened. When you have antibodies in your body and they encounter that antigen to which they were made, that's how fast those antibodies are protecting you. Within minutes, those antibodies are coming out. All right. All right. So we saw this. No reaction in the A, no reaction in the B, because there's no clumping. This is what clumping looks like. What blood type would this person be if they don't have a reaction here and here? They're O. And what are they for their Rh status? Positive, they're O positive, and that's the most common blood type. All right. Agglutination, antibodies doing their job. Okay. So more about B cells. Now I'm looking at the time here. And this is in the way, sorry. See if I can move it more. Okay. B cells activated by helper T cells. They produce the antibodies. And that production of antibodies, like I said before, is called antibody mediated or specific immunity or adaptive immunity or humoral immunity. That's 4 different ways that this type of immunity is called. Okay. Oop. Not advancing here. All right. Exposure of the B cell with its antibodies to the pathogen, chucks those antibodies at it, and it causes agglutination. All right. So here's something else that I'm adding to it. Plasma B cells, memory B cells, okay? I don't need to repeat this. I already did it on the previous slide, okay? So I don't need to do that. But here's something new that happens. This is the vaccine, why you need the boosters. When these B cells are exposed to the antigen for which they have antibodies prepared, right? You get the agglutination. What you also get is this B cell will clonally replicate itself logarithmically. So one B cell becomes two, two become four, four become eight, and it goes on and on and on like that. So that's huge protection right there. Okay, so I don't know if I have a effective antibodies. Okay, yeah, agglutination reaction. Let me move this here. Move. Okay, so agglutination reaction, antibodies stick to the antigen, clumping them all together so that other immune cells recognize them and destroy them. That's really the effect of antibodies. Okay. All right, so I want to get to one image and I'll come back to it. So here is the clonal replication, but I want to show you something here in a second. And then I'll come back to this on Friday. So I'm jumping ahead a lot. I'm jumping to this slide where it says vaccination, your first immune response, your booster, your shot number one, okay? Here is weeks after exposure to an antigen, all right? Your primary response in blue, all right? Primary response, you are exposed to an antigen, either naturally, you're exposed to a sick person, or you're given a vaccination. And usually vaccinations are for viruses, but not always. So now your immune system goes through the whole innate immunity, all those steps, and then a helper T cell gets a hold of that antigen from an APC and presents it to a B cell. And that's where we are. Memory and plasma B cells after your first exposure, your first vaccine, or whether it's a natural exposure. So you're exposed, let's say it's a vaccination. Your primary response is this. Look at that. Week 2, week 3, we start to get a good antibody concentration, okay? But notice it's only about halfway. It's only about halfway protective and depends, all right? And then that number of antibodies will wane, okay, a little bit. Your memory B cells stay there, but your plasma B cells numbers go down. Once you get your secondary exposure, so here's what I got. I'm a Lord of the Rings fan, okay? Here we have, and I wish I could come back to it here. We don't have it. It's not active, but these are like 4 little hobbits. They're on Hilltop, and then the ring wraiths found them. And so this is your primary response. You've got some plasma B cells and maybe some memory B cells are waiting in the wings, but you are outnumbered by the pathogens. Your protection is so-so. Once you get the secondary response, secondary vaccine, your plasma antibody levels are high. Your memory B cells come out of the wings. They were formed from the first vaccine, but once you expose the body to a second booster, those memory cells come out in force, and they're huge. They're like this. They're like an army like this, okay? Versus 4 little hobbits on Hilltop. That second vaccine, that's what's protecting you. This right here is much better than this right there. So I wanted to explain that because as nurses, you're going to have people that are resistant to vaccines. There's vaccine deniers. There's people who are anti-vaxxers. We'll get to that conversation on Friday, but you have to be the one who explains to them and try and convinces them, look, this is how it's protecting you, okay? So anyway, I'll come back to that on Friday, but I did want to explain this. Why do you get the first vaccine? And then why is the second vaccine important? And then we're in the era right now, we're talking about 3rd and 4th vaccines. So that's why. That's why. All right. I'll see you on Friday. You too.